welcome everyone to the Arthritis National Research Foundation's Researcher Spotlight webinar series. My name is Ashley Darrow. I'm the Director of Individual Giving here at the ANRF, and on behalf of our Board of Directors, the Scientific Advisory Board, and staff, I would like to welcome you all to our Spotlight on Lupus. We are so excited to have so many of you joining us from all around the world. Before we get started, I just want to share a couple of logistical pieces of information. Number one, this webinar is being recorded, and we're going to have it available on our website and YouTube channel in the coming weeks. Two, if you have any questions during the webinar, please type them into the Q&A box. If we aren't able to get to yours during the session, please email info@curearthritis.org after the webinar with your question. All right, and now to kick us off, we would love to hear from you. You should be seeing a poll on your screen right now, and we're just wanting to ask you what brings you here today. Are you living with lupus, arthritis? Are you a caregiver or a researcher? Be sure to put that in so we can see who's here with us today. All right, I will give you guys just a few seconds to keep going. Results are coming in so far, and it's looking like most people on the webinar are patients living with lupus. So that's a great place for us to begin. And what we'll learn today will help us sort of understand what's coming up next in terms of diagnosis and treatment. All right, and now I am honored to turn the mic over to our patient speaker, Erica Reyna. Erica is a married mom of 3 who works in fundraising and philanthropy at UT Health San Antonio and is a lupus advocate. She uses her lived experience to champion patient voices in research and policy. Erica serves on the Lupus Voices Council and as a peer educator for the Patient Advocates for Lupus Studies program, where she mentors patients and supports clinical trial diversity. Without further ado, I would like to welcome Erica to the stage.
Hi, everybody. So nice to meet everyone, and I'm glad that you guys are here today. I wanted to start from the beginning of my diagnosis story. So at first, I was just really tired all the time. I was a busy new mom. I was going to school, and I brought up my concerns, and I was told, "Oh, that's normal. You're a new mom, you know." So everyone said that's to be expected. And then came hair loss, and that was, oh, it's because you're a new mom and you're breastfeeding. The fatigue was horrible and unimaginable. I just slept all the time. I always felt like I was coming down with the flu. Joint pain was horrible. Brain fog was horrible. I would be speaking to somebody and then forget what word to use for like an apple or something, it just took me, I'm like, I would use thingamabob. What is that thingamabob? All the time, because I just could not remember. And yet for years, every doctor would say, you're just really stressed out. Again, you're a new mom, you're going to school, you have a lot of things going on. Hair loss is normal. Your wrists are in pain because you're typing all the time in undergrad. And then some would even say, like, maybe you need to go see a psychiatrist. This might be in your mind. And then I started really questioning myself. And I'm like, am I just like a weak person that can't handle being a mom now? Maybe I'm imagining this, like, am I going crazy, And so that went on for 7 years. And so at this point, I'm starting grad school, and I'm pushing through. And finally, the truth came after I was sent to an endocrinologist for some additional testing. So at the end of everything, I was diagnosed with 6 different autoimmune diseases. So lupus is the big one, Sjogren's, RA, pernicious anemia, dysautonomia, and then most recently, Hashimoto's. I feel like every time I go in, it's something else added on. But I never forget, there was one particular moment, I'm 27 at the time, just sitting there, listening to these lists of condition that sounded way bigger than me, right? My dream was at that time to go to nursing school. So I was doing all my prereqs to take care of patients. That's what I always wanted to do, but I was too sick to carry a full load of in-person nursing school, right? So that dream was shattered. And I remember collapsing. I'm going to try not to cry. I was collapsing on the floor in the restroom, sobbing. I'm like, my life is never going to be the same. All of these big aspirations that I had for myself, being very type A, very go-getter, felt like this distant memory where nothing is going to come to fruition now, And so I thought my career is going to be over before I even start this. And I thought I'd never work again. No one's going to want to hire me with all these doctor's appointments that I have to go to. And as a young mom, that really broke me. I'm worried about what kind of future my kid's going to have with a mom that's always in bed and always in pain all the time, right? And that's when something really unexpected happened. I started speaking to policymakers, and it just started little by little. And I was so nervous. I'm like, I cannot do this, but I thought, like, I have to for my kids. I'm gonna do this. I'm gonna ask for funding for more research. And so I really found purpose in the very thing that almost took me down. I started just sharing my story in small ways on my Instagram page with that hardly had any followers, on Facebook groups with other lupus patients. And then occasionally I would get DMs and people would say, oh my gosh, my sister's having some of these symptoms, or my cousin's having some of these symptoms. And these late night talks with patients who were just as scared and worried and overwhelmed as I would be were slowly turning into something bigger. So today I serve on a couple of different ways in my advocacy. So I serve on the Lupus Voices Council, and I'm also a peer educator with the PALS program with the Lupus Research Alliance. And so I do things like sitting on panels like this. I've met with pharmaceutical leaders, policymakers, mostly pushing for more inclusive trials and just better care overall. I fight for people of color specifically to be represented in research. Often the times because we're excluded a lot, and we're one of the groups that is the most highly affected by these things. So today, lupus is still a constant battle. There's ups and downs all the time. I wake up in the morning and it's kind of like checking the weather, but with your body, I'm like, is it going to be rainy or sunshine today? Like, how is this day going to go? And there are some days where I have to cancel plans. I can't do it. So I'll slow down, have a moment of grieving that I can't do something that I really wanted to do, but I just keep going. I do it for my kids and for other patients and that younger version of me who thought her life was over at 27, right? So I think if there's one thing that you can take from this story is that chronic illness does not make you a fragile person. It can make you super fierce, and it took the future away of what I thought my life was going to be like, but it really gave me a voice and a big fire and a purpose that I never expected would come from all of these things.
Thank you so much for sharing your story, Erica. I love leading with fierceness. That's an incredible message for everyone listening today. We are going to be hearing from Erica again later in the webinar in our Q&A. So be sure to add your questions if you have any for her in the Q&A box, and we'll try to get to as many as we can. Now we are going to hear from Dr. Tarin Bigley, who is a pediatric rheumatologist and researcher who is passionate about understanding how viruses may trigger autoimmune diseases and affect the immune system. His work focuses on how certain viral infections may play a role in conditions such as autoimmunity and neuroinflammation. Outside the lab, he enjoys an active life with his wife, two children, spending time outdoors, traveling, and staying active through running and biking. I'll kick it off to you.
All right. Thanks, Ashley, and thanks, Erica, for sharing this story. It's actually probably a really nice segue into most of lupus researchers' focus, but definitely one into mine. And um, as Ashley mentioned, I'm really interested in how viruses might act as a trigger for autoimmune diseases like lupus. We know that autoimmune diseases are probably multifactorial in terms of their cause, but we often don't know the cause, especially by the time we as rheumatologists, hopefully early in disease, see somebody. But as some of you know, it can be months to years after maybe you started having symptoms. And we think about this idea that the difference between an autoimmune disease and an infectious disease is that in the latter, a pathogen, an infectious agent has been identified. We know this isn't completely true, and as a pediatrician, I actually see clearly genetic causes of autoimmune disease. But for most people, that a single gene causing disease is not the case. And so I actually want to bring up two cases to kind of illustrate um, some of the challenges we have in diagnosing lupus. And so I'm going to use pediatric cases, but these are not that different than what might be seen in an adult case. And so the first case is a 14-year-old female, previously healthy, and developed severe face rash and joint pain after prolonged sun exposure. Um, saw saw someone who sent some labs, which were mostly normal, but had a uh, high titer or high value of anti-nuclear antibody or ANA. And I think most people in this group are familiar with this. It is an antibody that our immune cell makes that recognizes human cells. But we know as rheumatologists that lots of people who don't have lupus or other autoimmune diseases can have these antibodies. So once they come to my clinic, other than this kind of fading rash, the exam is normal. And in this case, what do we do? How concerned do we need to be that this is lupus or this is not lupus? And how concerned do we need to be about this antibody level? Is this someone who maybe is going to develop lupus, or do we not need to be concerned about that? And we can provide reassurance and say this is probably just a bad sunburn, and sunburn so bad that you had some inflammation associated with it. And this is in contrast to our next case, also a previously healthy 14-year-old, developed this severe facial rash and joint pain associated with some sun exposure. Uh, Labs were obtained, also showed very similar value of ANA, but in this patient, they had low blood counts like anemia. Their liver enzymes were elevated, suggesting maybe some liver inflammation. And they had other antibodies and markers of inflammation that were elevated. And we see them on exam. They have this rash, they have some joint tenderness. I think we as rheumatologists would recognize this pretty quickly as lupus. We would be really concerned. There might be some other tests we do, but I think all of us would be considering treatment at this time. But I think this illustrates how um, different cases of lupus might present to our clinic, but also some of the conundrum in trying to diagnose lupus and differentiate those that are clearly lupus versus those that may be predisposed but haven't quite reached what we would call like a threshold for diagnosis. So let's go back to my research. I think within the field, a lot of us have suspected viruses can contribute to the onset or maybe flare of lupus. Um, but how they contribute to the transition from health to autoimmunity is really difficult to study. Evidence is pretty limited, and that's because all of us are being infected with different viruses or bacteria throughout our lives. Those of us that have kids know that, right now is the time that we're seeing, 10 different viruses a day that our kids are bringing home. And we don't, it's really hard to understand how the timing of infection is contributing to disease. The virus that maybe contributed to disease may not be present still by the time I, as a rheumatologist, see someone in clinic. We don't usually know the infection status. Is this the first time they've seen a virus? Is this a common childhood virus that maybe occurred years prior to onset of disease? One of the things that we do know is that patients with lupus show signs of immune dysregulation, immune problems months to years before diagnosis. And this is just showing you a study from a military cohort where they had blood samples and they found that compared to time zero, which is when people were diagnosed with lupus, these autoantibodies, things like an ANA or a double-stranded DNA that we commonly use to help diagnose lupus, these were present years before the actual diagnosis. And this is something that has been seen in multiple autoimmune diseases that if you look months to years before the actual diagnosis here in the yellow of, of disease, there are immune disturbances and autoantibodies present long before that. And we don't understand why they're present long before that and what's really causing these changes, this progression to autoimmune disease. We know that there are genetic factors that contribute, but even in identical twins, if one twin has lupus, there's only about a 25 to 40% chance the other twin will get lupus. So there are clearly environmental factors that are contributing, and probably there are multiple hits throughout life that are causing disease. And so, how do we look at these environmental contributors that are occurring long before I, as a rheumatologist, will actually see them in clinic? And so that's what my lab's focused on. And I'm going to go through some data we've collected, but I will explain some of this in enough detail to really understand what we're looking at. So one of the viruses we're studying, amongst others, are these viruses called roseola viruses. The official name for these are human herpes virus or HHV6 and 7. These are really common childhood infections, and I think most parents have heard of this. You know, your kid sometime between age 6 months and 3 years gets a fever and then breaks out in a rash, and the fever goes away. And maybe you see your doctor and they say your kid had roseola, but they're going to be fine. This is a self-limited infection. And this happens to most kids. So by the time the kids are school age, greater than 90% have seen this virus. And it's been linked to multiple autoimmune conditions. But how do you study a virus in humans that is infecting really early during childhood when the autoimmune disease we're seeing is occurring much later in life? It's really hard to control because healthy people and people with lupus have had this virus. And this virus also stays with you the rest of your life. It's not thought to cause a lot of disease, but it's present in our bodies forever. And so what we've done is we've utilized animal models. And I want to talk a little bit about one of the types of cells that contributes to autoimmune disease, and that's T cells. And I'm not going to go into a lot of immunology on this, but what we know is that in general, what's happening from early in life through later in life is that T cells are trained. And this is good. And they're trained in an organ called the thymus, which is right above your heart. And what happens is any T cell that recognizes your own tissue is removed. And this is a really good thing because we don't want T cells that recognize our own body to go out into the body and cause disease. And so the T cells that are trained and are able to go into the body are ideally the ones that can recognize viruses and bacteria and help us stay healthy. And they're not the ones that would cause disease, cause autoimmune disease. And so the question we're asking is, what if you have a virus that can infect that organ, the thymus, that trains the T cells? Would this cause T cells that can recognize your own body? And what we found in our studies is that this roseola virus, when it infects really early in life, despite virus only being detected early in life, is resulting in development of autoimmune disease later in life. And Erica, I thought of you when you were talking about your story. What's really interesting is these mice develop autoimmune gastritis. So what's associated with pernicious anemia. And they develop inflammation in their stomach. We see that basically all the mice that get this virus really early in life develop disease. They develop these autoantibodies. And just like that graph that I showed you previously, they develop autoantibodies before they develop disease. What I will tell you too is that the timing of infection really matters. And I'm not showing this data, but if we infect later in life, they don't go on to develop disease. And so that gives us a little bit of idea what's going on. But what was also really interesting is despite the fact that they have disease that's pretty limited to a single organ. And these mice overall look pretty healthy. They have a lot of autoantibodies that are associated with lupus and other related autoimmune disease. And this is compared to our uninfected controls or mice that were treated with the antiviral. So we really wanted to try to understand, why do they have all these autoantibodies that we associate with things like lupus, but they have pretty limited disease? And we wondered if these mice, which are kept in really clean conditions, they're not seeing other viruses, aren't really a good representation of humans in this case, where we're seeing viruses, our immune system is being bombarded all the time with stimuli. And so we wanted to know is, if we add additional immune stimulation, could we induce autoimmune disease very similar to this graph I showed you, where maybe our mouse model is kind of in this in-between stage where they have a little bit of autoimmune disease, but they need additional environmental hits to develop systemic disease like lupus. And so our first environmental hit is our virus. But then what we used is we used a molecule that looks a lot like looks a lot like a virus. Our immune system detects this as a virus. This is called R848 or resiquimod. And what if we give this multiple times in adulthood and evaluate whether or not they develop worse autoimmune disease? And I apologize, this is the only slide that did not transition well from Mac to PC. But what we see is, I want you to just focus on the green, which is our mice that receive the virus and then receive the immune stimulation later in life, this R848 chemical, is they develop low blood cell levels, which is very common to what we see in lupus or some other autoimmune disease. They develop a higher ANA level, and they develop a high double-stranded DNA antibody level. But what's even more pronounced is when we look at the tissue, we see all these inflammatory cells in organs like the kidney, the heart, the liver. We also see it in the lung, the spleen, and the salivary gland. So these organs that we associate with our systemic autoimmune diseases like lupus or related diseases. And what's really interesting when we kind of look closer at the immune system, and this is not to show these specific immune proteins, but just to show that the response to the inflammation is different in the mice that were infected and then received immune stimulation later, with some of these immune proteins going up, while others not responding to the same stimulus as you might see in an uninfected mice. That this early life viral infection is changing to a very molecular level how the immune system is responding to subsequent immune stimulation later in life. And what we even see is, I showed you that blood cell levels go down. So the white blood cell levels here in the green are decreased compared to our controls. But when we look in the spleen, which is, I think most people are familiar with, is another immune organ, those cells seem to be increasing in the spleen. So they're actually changing where they're going. We're seeing them not in the blood, but instead in other organs. And so the last thing I just want to show you is this question we had, which is this immune stimulation just causing our virus to reactivate? Is that what's causing disease? Has the immune system changed to the point that when you see other stimulus, when your immune system is really challenged, viruses reactivate and they cause more inflammation? So what we did is, at this time in adulthood that we were stimulating the immune system, we gave the mice an antiviral. And this is an antiviral that we use not uncommonly in the clinic, called valganciclovir, or VGCV. And what we saw is that if you look at the green bars, which were the antiviral treatment compared to the mice that weren't treated with antiviral, this did not reduce disease. So it doesn't appear that the virus is reactivating and causing disease, but rather that the immune system has been so altered by that early infection that this repeated stimulation is what is causing disease. And so I'm going to end there in terms of just showing some data on what we're working on. But our lab is really trying to utilize these animal models as well as human studies to understand this kind of walk throughout life from health to autoimmune disease, where an early life infection at certain times in a certain background causes the immune system to be dysregulated, to start to cause autoimmune disease, but also a recognition that additional immune stimulation is needed beyond that. So it's really causing a predisposition to disease. And we're trying to understand what this can tell us about, our case one, where they have a high antibody level, but we don't know yet if they're going to go on to develop autoimmune disease. They might have some vague symptoms that don't really fit yet within our diagnosis of autoimmune disease. And also to understand whether or not we can identify what's going on early in life, and there's a signature of these, early life infections or other disturbances that tell us which patients maybe are the ones that are predisposed and, have a lot of fatigue, but haven't yet developed other symptoms that make their diagnosis clear. So I want to just quickly thank my lab. There are multiple people that have worked on this, including some of our collaborators, but also give a shout out to the ANRF who they funded this work. I think it's important for people with lupus to know that this funding is instrumental for a lot of us in getting some of these studies going, especially if they are pretty early on or exploratory for some of us that are really early in our career. But the NRF also sets up networks for us as researchers and physicians to get together, to share resources, to share our stories, to meet patients, and it really is doing amazing things. So I will leave it at that and hopefully answer some questions later in our panel discussion.
Thank you so much, Dr. Bigley. That was really exciting to hear about. To your point, please put questions in the Q&A box if you have them for either of our researchers or our patient partner today. Now I have the pleasure of introducing Dr. Sue Wacleche, a lupus researcher and assistant professor at Penn State, inspired in part by her great uncle's lupus diagnosis and her commitment to improving health outcomes for women of color who are disproportionately affected by this disease. Her research focuses on uncovering how certain immune pathways drive lupus, paving the way for potential new treatments. She is dedicated to giving patients hope by advancing discoveries that can lead to better therapies and improved quality of life. I'll kick it off to you.
Thank you so much, Ashley, for this warm introduction. I want to start by talking a little bit about myself. So I was born in Canada. I am first generation, so I'm the first of my family who got a college degree, first who got a bachelor, master, and PhD. And then I decided to go all the way to the top. So I'm a scientist purely, I'm not a clinician. And I started my scientific journey by performing a bachelor studies in microbiology and immunology at McGill University. And this is where I found my passion for immunology. And then afterwards, I decided to pursue my studies into doing a master's and a PhD at the University of Montreal. I was born and raised in Montreal. And I've studied T cells in the context of HIV. And then at the end of my PhD, I decided that I wasn't a disease person, but I was an immunologist. And I decided to, after my PhD, to leave the world of HIV and infectious disease and to leave my beautiful country, Canada. And I met this outstanding rheumatologist called Dr. Deepak Rao at Brigham Women's Hospital and Harvard Medical School. And he gave me this project to understand more uh, T cells, subsets of T cells, and I'll talk to you about that. And my postdoctoral studies at the laboratory of Dr. Rao changed me forever because I am an own investigator, and my research is about lupus. Well, it's about understanding the different cellular players contributing to autoimmunity and how autoimmunity is induced, and we're using lupus as a model. And we want to understand what are the pathogenic meaning the cells that are harmful, that are going to contribute to the disease, and then the cells that are non-pathogenic, that is going to protect. So cells that is going to help get back to a normal balance. So autoimmunity is about an unbalance. It's where the immune system is not balanced at all. And there's a disbalance that the body cannot reach. It can't reach out the normal balance anymore. And they develop a new homeostasis or a new balance, which is not really the balance that healthy people has. And there are some cells contributing to this disbalance, and cells that are contributing to get back to the normal balance. And this is what my lab is all about, studying those cells. So just to let you know how we are in my lab, I'm all about humans. So I've done all my studies in HIV, and when I was a graduate student, I didn't have any animal models. The only animal models that at the time that was available for HIV was macaque, or the primates, the monkeys, and our lab didn't have the funding to have access to this resource, so we relied more on blood. And when I came to Boston, it was the same. I did my studies using tissues or blood or serum samples. So I am still continuing this path. So I'm a human immunologist. I only care about the humans. I don't really care about the animals. And what we do is that we're going to, in our lab, we recruit either healthy donors or lupus patients or rheumatoid arthritis patients. We take their blood samples, and then we're going to do some ex vivo experiments. What we want to understand is what is the difference between the healthy individual, the cells, the population that is expressed on the healthy individuals, and those that are expressed in lupus. We try to find the differences because we are telling ourselves in our lab that probably the differences that we observe may help us better understand the disease and may help us better to find a therapeutic strategy. So we start by our observation by doing multiple experiments, whether using blood, isolating white blood cells, or using serum and looking at inflammatory cytokine production. We also do a series of experiments in vitro that I'm not going to go into details, but doing research on humans, we have limitations. And that's the reason why in my lab, we're going to use mice model, which will allow for us to understand the physiological mechanism of our observation that we see over here in humans. So like I said, what I'm interested in is observing what is the anomalies, what is the difference that we're seeing between healthy individuals and lupus patients and doing a series of experiments, whether it's in vitro, working with human cells, or in the mice, just understanding in a physiologically environment, understanding our primary observations. Okay, so just talk a little bit about lupus. But as you might know about it, I just want to pinpoint that lupus, it affects 3.41 millions worldwide. So it's quite significant, and it affects, just like Erica mentioned, it affects more, nine times more women compared to men. That's my next point, but. Every year, unfortunately, out of 100,000 people, about 5 people will be diagnosed with lupus. And just like I mentioned, it affects 9 times more women compared to men, and it affects 3 times more Black individuals or people of color compared to Caucasians. And that, it changed my life, honestly, when I realized that I was working in a disease that affected my community. I am a Black woman, and knowing also at the time where I was actually deciding what kind of scientist that I wanted to become, I learned that my great uncle was diagnosed with lupus after 3 years of hell. Every year I would see my great uncle. I didn't understand him, and finally the diagnosis came, and combined to his diagnosis and the fact that I realized that it affects more my community in general, I decided to continue my career in working in lupus. So from an immunological point of view, I'm going to try to spare you the details, but lupus is characterized with this aggregation of this recruitment of those T cells and B cells, and those, just like Dr. Bigley tried to let you know, the T cells and the B cells, both of them, they are aimed to protect against pathogens, just like viruses or bacteria. But in the case of lupus, they actually are reacting to protein from the cell. So protein produced by the body. And the T cells will help the B cells mature and then producing those antibodies. And it's those antibodies that is going to lead to the development of lupus. And lupus is also about organ damages. So there is unfortunately only 2 biologics that has been approved by the FDA for specific treatment of lupus, and these are anifrolumab and belimumab. And I'll talk a little bit more on our research that we have performed with anifrolumab, which is an anti-type 1 interferon receptor. So lupus, one of the hallmark is the fact that patients, they're going to produce a lot of this pro-inflammatory cytokines that is called type 1 interferon, and this will lead to organ damage. Okay, so what in my research as a postdoc in Dr. Rao's lab, what we've learned is that a T cell is going to produce a factor called CXCL13. CXCL13 is going to tell the B cell, come in my vicinity, let's hang out. What's going to happen is that there's going to be an immune reaction. And this immune reaction, the B cells are going to be able to develop, mature, and producing amounts of autoantibodies, which will lead to the development of lupus. So what we've learned using anifrolumab, so we could follow patients over a year with anifrolumab, and these patients were stratified into 2 groups, interferon high and interferon low. So interferon high group are a group of patients that's going to react more to interferon. And interferon low is this is going to be patient that's not going to react, their cells are not going to react to interferon. And what we have discovered is that the CXCR13 factor that I just spoke, well, when that will allow the migration, the recruitment of those B cells when the patient gets treated over time with anifrolumab, only in those interferon high patient group, there's going to be a decrease of this chemokine. Now, for this factor, we don't know why it only affects this group of patients and not this group. But this highlights the fact that lupus is a very complex disease, and that the therapies that it's not going to be it's not going to be specifically for all work in all patients. But for lupus, I guess we will need a tailored therapy, tailored drugs or therapies, because it's so heterogeneous, and the way that someone will receive a treatment and the benefits of it, it might not apply to another person. So we wondered whether the decrease in this CXCL13 over time, was it due to a decreased percentage of the cells that was producing those CXCL13? And we did a series of transcriptomic analysis. I'm not going to go through it, but we've actually shown that, and we've used blood. We've collected patients before and pre-anifrolumab treatment and after anifrolumab treatment. So on the blue dots are the patients, the frequency of CD4 T cells, their frequency before administration of anifrolumab, and then the orange triangle is their percentage of memory CD4 T cells after anifrolumab, and we're looking at the TPH cells, and these are the cells that produce CXCL13, this factor that I was talking about, and we've shown that there is a decrease of these cells producing CXCL13, and this is a way. It's a hope. It gives us hope that anifrolumab will lead to a more the balance, a rebalance of the balance that we're hoping that lupus patient can reach a similar balance that that healthy individuals are having. So in this, with this, I'd like to thank. Oh, and also the reason why I'm so sorry. This is the most important slide, by the way. Well, the second most, the last one is my acknowledgement. I do research just like Dr. Bigley to help patients and to provide therapeutic strategies that will at least make their life better. And we do research to heal, but at least for them to have a better quality of life. And to this, I am going to acknowledge, and I want to, it's my most important slide because I want to acknowledge the patients for your gift. If you don't give blood, scientists just like myself, we're not able to do research. So thank you so much for the time of you coming into the clinics and giving your blood. Thank you.
Thank you so much, Dr. Wacleche. It was really interesting to hear more about your work. I'm sure folks have lots of questions, but before we open up for the Q&A, we'd love to hear from the folks on this call just one more time. We want to know how lupus impacts, we know it impacts people in so many different ways, and we'd like to know what's the number one thing you'd like to better understand. So check the poll out on your screen and go ahead and choose one that resonates most with you. Is it symptoms and flare patterns, diagnosis and testing, research in the future? Which of the options that you see there are you most interested in? So I'll give you guys just a few seconds to answer that. All right, great. Thank you. We're seeing some great answers coming in. Lots of folks wanting to learn more about research, the future, diagnosis and testing. Thank you so much for sharing. So we've got a few questions that have been coming in that really touch on a lot of these areas. So let's go ahead and dive right into the Q&A, and we'll be able to hear more from our speakers. All right, so it looks like the first question we have is for Erica. Erica, what advice would you give to someone who's newly diagnosed and feeling overwhelmed?
Oh gosh, I think the thing that I learned to do is just to give myself grace and to make sure that I had a community in place. I think I was really worried about asking for help, or like I thought people would be judgmental. Same thing with work. I was really worried, oh gosh, if I tell my employer, they're going to fire me and I'm not going to have a job anymore. So I think just give yourself grace. Is actually letting people know about it has opened more doors and closed them. So yeah, grace, and don't be scared to ask for help. I like soccer moms. They're more than willing to help me out to pick up my kids. It's been amazing.
That's great advice. Thank you for sharing. Our next questions is for both of our researchers, Drs. Bigley and Wacleche. What do you think are the most promising discoveries on the horizon for lupus treatment? I'll go first on my screen. Sue.
Oh, okay. Well, there is and Dr. Bigley, please tell me what do you think about this. But of course, there's the CAR T cells. It's starting to be very fashion in the world of lupus, but also there's a lot of controversies. But this is a promising avenue. So engineering T cells to target the B cells. So that's a promising avenue. Dr. Bigley?
Yeah, I mean, I think that's the hottest topic right now in lupus. And one of the real challenges with that is which patients are we going to use that for, which aren't. And so I can kind of explain for anyone who hasn't heard of this. Basically, what has been researched recently is the use of these cells called CAR T cells. These are the T cells that we talked about that in our bodies normally we want fighting infection, but we can engineer these T cells to fight those autoimmune, in this case, B cells. And when we give people this therapy, it's removing these cells from the body. And we already do this in some fashion for anyone who's received Rituxan or rituximab. That medication actually is targeting the same cells, but for reasons we don't completely understand yet, or maybe incompletely understand, the cellular therapy seems to, in at least in the small studies we have, seems to be more effective. And I think everyone is really excited about it because we can potentially target those immune cells. I think the other thing, from a research perspective, is, and I didn't actually talk about any of my human work, but I know my lab, as well as others, are really trying to stratify or separate different lupus populations. We know clinically some people, some people come to clinic predominantly with arthritis and rash and low blood counts, while other people have more kidney disease or liver disease. And the technology in research has advanced so much. So the human studies that I actually didn't talk about are really characterizing lots of different parts of the immune cells, and that like very fine analysis of the immune cells is telling us different things about populations within the lupus populations. And for example, I see pediatric patients, and what's really interesting is the younger you get, the less of a female to male difference there is. And so maybe certain groups of lupus need different diagnostics or different treatments. And I think that we're still probably 5+ years on the horizon from really understanding all of that data, but I think it will change how we approach diagnostics and treatment.
Thank you for that. It looks like the next question is also for you, Dr. Bigley.
Okay.
How does your research translate to patients, and what would that look like in a human trial?
Yeah, I it's a challenging question. One of the reasons we're using a mouse model is because the we can control we can control when infection occurs really well. We can do studies over time that we couldn't do in in people. For example, a lot of these viruses don't infect mice, and so we have to use a similar virus in mice, but also, um, you know, like Erica, for you, for example, no one probably was looking to get blood samples early on in disease. So we often just don't have that data of what's happening months to years before actual diagnosis. And so a really good question is, how do we translate this? I think one of them is identifying um, identifying some of these factors in an infection that are contributing to disease. And so, for example, we can now start looking in certain patient populations, and this is research that we're actively doing in the lab, that might be more susceptible to the effects of these viruses. And we can now start to compare the molecular experimental data we have collected in mice to humans. Is there an immunologic signature that characterizes this predisposition to disease? And one of the really cool things um, in in rheumatology cohorts, research cohorts, is that we are actually starting to collect more samples over time. So we can go back, people like me can go back and make those comparisons. And so I think that is one of the kind of translational components. The other thing we're looking at, and I showed you some of those immune system changes that seem to be characteristic of that early life viral infection. We're now looking in human samples to see if the same thing is going on in those patients that have really high levels of autoantibodies but haven't yet been diagnosed. And if that's the case, those might be really good markers of transition to disease.
Thank you for that. We have another question for Erica. How has your experience with treatment options evolved over time? Have you noticed a shift in what's available to you?
I started off with Plaquenil just like everyone else. I've been on steroids for longer than I can remember. I've tried various biologics, and then eventually I'm not responding anymore, so I get transferred to something else. I did start IVIG therapy, and that seems to have helped tremendously. But yes, CAR T therapy is what the talks have been about recently that I've gone to, and it sounds really great and promising. So I'm super, super excited. I think the most people though that I've talked to have had a hard time getting into trials because maybe they don't have nephritis, but they're still feeling sick, or maybe there's not a location close to where it's at and they can't afford to travel and all these different things. And so that's what a lot of the advocacy work that we're doing is kind of focusing on, is making things more accessible to patients, and just kind of spreading awareness so they even know that these things are out there.
Yeah, great. Okay, so we have time for one last question, and this is for everyone. So if you could fast forward 10 years, what's one change you'd hope to see in how lupus is treated or managed? I'll kick it off to Dr. Wacleche first.
Um, well, I guess having better diagnosis, that would be great. Um, I mean, knowledge is power. More knowledge, more stratification of the patients, that will help us better understand the disease. I think that would be my response.
Thank you. Dr. Bickley?
Yeah. I mean, I just seeing patients and everything they go through, I think doing a better job of early diagnosis and maybe early treatment would be great. Um, I would I would love to imagine a world in 10 years where we have a treatment that's curative. I don't know that we'll get there, but that's what all of us are working towards, that we have really good treatments. And even if they're maybe not curative, they help control disease really well. Because any of us that see patients with lupus or know people with lupus understand that unfortunately the disease is really dynamic and response to treatment can last for months or years for people. Flares are not uncommon, and I hope, and a lot of the medications have side effects that we're constantly, as a patient physician team are trying to manage. So I would love that in the next 10 years we get there, and I think uh, we are working really hard to do that.
Thank you. And Erica, you're up
I think
last.
I'd like to just see more patients and doctors, and I've been fortunate enough to have a team of doctors that work in tandem with me together as a team. So I'd like to see more doctors open to kind of like an equal partnership. You know the science, but I know my body, and just being listened to. I think that's super important. Also,
Absolutely.
mental health stuff. I have a lot of people that are diagnosed, and one of the first things I always say is, because my doctor did that, please make an appointment with a mental health professional. I think that made a huge difference when I was first diagnosed, and how I lived the rest of my life going to therapy and things like that.
Great
Yeah,
advice. Thank you for
maybe
sharing.
I was just going to add really quick too that um, kind of that point Erica made about, team and
Yep.
team involvement, and that it really does have to be a partnership. I think more and more places are recognizing this.
Hmm.
Even at WashU, it's very recently we have put together a team so we can start doing this so our patients can see us and nephrology and dermatology and adolescent medicine and have access to social work and psychiatry, because lupus can affect so many
Hmm.
aspects of people's lives. And I think over the next 10 years, it would be great if more people had access to those kind of clinics.
Yeah.
And also, I just want to add that I'd like also to see in 10 years or even more 5 years, um, more people of color participating in clinical
Yes.
trials. That's very important because the disease affects more people of color than Caucasians. Why is Caucasians are participating more than people of color? That's because people of color are skeptic
Hmm.
and hoping that they're seeing a person of color working in lupus. Hoping I'm really hoping that this will give them a little bit of hope that yes, they can, and give them motivation to participate in clinical trials, because what's the point of having a population, trying those clinical trials, but not the population that is affected the most. So
Hmm.
there's a problem in that, and hopefully that will change over time.
Hmm.
Yeah.
Absolutely. Thank you for that. Before we wrap up, I want to turn the mic back over to Erica just one more time to share some exciting resources from Lupus Therapeutics.
Yes, I'm so excited about it. So I briefly wanted to speak about some important work that Lupus Therapeutics is doing with their PALS program. So PALS stands for Patient Advocates for Lupus Studies, and it's more of a peer education program co-designed with people that actually have lupus or caregivers of people with lupus. And the goal of this program is to improve knowledge, awareness, engagement, and retention in these lupus clinical trials. So to do this, patient advocates called PALs will hold sessions with program participants, educating them about what clinical research looks like and why it's important. I did recently train as a PAL, and I'm looking forward to working with participants on these topics really soon. So Lupus Therapeutics is moving into a new phase of this program. We're implementing two components. So one is related to educating individuals on clinical research participation, and the other thing is PALS offering support during specific lupus clinical trials as they go along. And we're really looking forward to this next chapter, as well as expanding not only the education, but also to support people that are living with lupus, which is so needed for more representation, like people of color in clinical trials.
Thank you so much for sharing that great resource. As we wrap up, I just wanted to say thank you so much, Erica, our fantastic patient speaker. We so appreciate you being here and sharing your story today. I also want to thank our wonderful researchers for sharing their innovative work towards understanding lupus. You can see firsthand the impact ANRF has had in supporting incredible individuals like these and their quest for a cure. We also want to thank our partner Lupus Therapeutics for co-sponsoring this event. And thank you to everyone who joined us for this great event. If you enjoyed this webinar, we kindly ask you to consider making a donation today by visiting curearthritis.org/donate. Every contribution helps and directly supports scientists like the ones you've heard from here today. We will be sending our live attendees a survey after this event. Your feedback really helps us find direction for future webinars, so please share your comments with us so we can continue providing important and relevant content to you. And please don't forget to sign up for our other researcher spotlight webinars. We'll be live at the same time on September 24th with our spondyloarthritis webinar, October 8th with a focus on Sjogren's disease, and October 22nd with rheumatoid arthritis. Thank you all so much for coming, and please remember to follow us on social media as well to stay connected. Have a great day.